So welcome to the afternoon session. This, in a lot of ways, maybe should have had this session first because everything starts with vaccine production with a virus in the laboratory and the characterization and the surveillance and epidemiology, and we don't get anywhere if we don't have the information we need on the viruses. So I think this will be a very good session on surveillance and laboratory support. And I would, in addition to having to have regulatory support for capacity building in countries, I think we also have to have adequate laboratory and surveillance support, as has already been mentioned. So we have two excellent speakers today. First I'll introduce is my former boss, Dr. Nancy Cox, who is, as you all know, Director of the Influenza Division at the U.S. CDC. So Nancy.
Thanks, Mike. I've just got 10 or 15 minutes to talk about what is a very, very big topic, and so I'll be just kind of skimming the surface and then making a couple of very modest suggestions about how we might improve the system, or what we might strive for to improve availability of vaccine at the time we really need it, with respect to epidemics and particularly with respect to pandemics. So my talk doesn't really have an outline, but it has themes. And they are challenges for global influenza surveillance and capacity building, the need for funding to continue this process, and the need for very, very good partnerships to make it sustainable. All of these things continue. And we have the ongoing need for more rapid detection of new pathogens. We did pretty well with this pandemic, but we have some areas where I think we could make clear improvements. And then there are the challenges for faster vaccine development and production. And I'll be saying perhaps a couple of things that I hope that Jerry will pick up on his presentation, but he and I really haven't had a great deal of a great opportunity to talk prior to today, so we'll perhaps have some of those elements to discuss after the presentations. And then I do want to mention that this pandemic has really demonstrated very, very clearly the importance of well-developed partnerships and rapid virus and information sharing. And I think that there's probably no one in this room that would disagree with that. So as we were preparing for the next pandemic, as many people have mentioned, the H5N1 experience really shaped our expectations. Not so much those of us who had been in the field for a very long time, but for many of the policymakers and many of the people who were involved in influenza only during the time that H5N1 posed a threat. So the expectation and most of the exercises and playbooks were really built on the assumption that the next pandemic would emerge in Asia, certainly not in North America. And I think that this pandemic has really emphasized what those of us in the influenza area for decades had known, and that is that we cannot predict influenza, and the next pandemic could emerge from anywhere in the world, and that's why we need global surveillance. The expectation was that the origin would be from avian species, not from swine, and that was based on the H5N1 experience. Those of us who had been in the influenza field for decades knew very well that swine had been postulated as a reservoir for novel influenza virus emergence and as a mixing vessel. And sure enough, that hypothesis was demonstrated to have a lot of merit. When we were planning for H5N1 scenarios, we really tended to look at the 1918 pandemic as a model, simply because the case fatality ratio for H5N1 was so high. And in any case, even if we hadn't been biased by the H5N1 experience, which perhaps wasn't such a bad thing, preparing global surveillance and diagnostic capability for a pandemic is challenging because the virus is really the problem. And a lot of times we don't really face the fact that influenza is an extremely challenging pathogen. And it is. And so we can think about all kinds of fancy ways to screen and put viruses in libraries and so on and so forth. But Mother Nature has a way of throwing things from left field right in our path, and we have to pick up the ball and run from there. So, as everybody knows, we have the different subtypes and constant evolution of the hemagglutinin neuraminidase and constant reassortment of genes, and that it's really difficult to predict. We have tried to predict what the next epidemic virus will look like, what the next pandemic virus will look like, and we do pretty well with epidemics, but certainly not so well with pandemics. Most of our diagnostic tests do not differentiate subtypes. That is a big problem. We need to know if viruses are unsubtypable with the current tests, and therefore they raise a red flag. And I think there's a lot more awareness of this globally than there was prior to the H1N1 experience. And I just want to I'll mention this a couple of times, I think there's a great need for rapid tests for antiviral resistance testing and for rapid immunity testing so that we know who's immune, who's already had flu, and who hasn't. Now, we were really prepared in the United States for the H1N1 emergence, both by design and by luck. We had improved our diagnostic testing at the state health department level by developing and deploying a 5-target real-time PCR assay that diagnosed influenza A, B, H1, H3, and H5. We'd done that in preparation for an H5 pandemic, and we had also designed the primers and probes such that if swine influenza viruses that were circulating in pigs in the U.S. infected humans, we would be able to differentiate those. They would come up as unsubtypables. We'd also made novel influenza virus infections of humans notifiable by the states. And so this sort of raised the awareness, up the ante, if you will. And so the states had to report to us and send to us specimens that were unsubtypable, or they were supposed to. And through all of these activities and growing awareness, we had seen increasing numbers of swine influenza cases detected during the past 5 years. And of course, most of you know, you've read these articles, but I really can't emphasize enough how these events really shaped our response, because these events had led to increasing efforts at the state level, at the national level, and in concert with our colleagues at USDA to identify and investigate human cases of swine influenza. We weren't always successful in doing the kinds of investigations that we would like. Often the event was over by the time the diagnostic test was actually finalized. And it was difficult to do retrospective investigations, but nevertheless, it certainly did raise awareness. In these cases, there was apparent limited secondary transmission, but these detections and the characterization of those viruses really led to increased awareness in states and better laboratory preparedness at CDC, because then we developed the primers probes for diagnosis of these viruses and the primers for rapid sequencing of the viruses. So that's basically what led to a very rapid timeline for testing and identifying the first clinical samples. And I won't go through this in detail, but needless to say, by April 17th, when we had the second specimen arrived, we'd already looked at partial sequences for 8 gene segments of the first virus, and we knew that the genes appeared, at least with these partial gene sequences, to be closest to those of influenza viruses previously detected in swine in North America, but that a couple of the genes were most closely related to swine influenza viruses identified in Europe and Asia. And this was a new reassortant virus. So we had, because we were prepared, we could do this analysis very, very quickly. By April 18th, we had sequenced all 8 gene segments of the first isolate, California 409. And then once we had that information and we're pretty sure that the second isolate was identical to it, we actually made a decision that we would clean our GLP lab to begin preparation of a reverse genetics vaccine candidate. And of course, you have to clean your lab, you have to have your biosafety group come in, and basically make sure that you have gotten rid of everything that you were working with before and that you could start on a new preparation of a new reverse genetics generated vaccine candidate. And then, of course, on April 23rd, all the information came together with our colleagues in Canada and particularly our colleagues in Mexico when we collectively realized that what had been going on in the U.S. was related to what was going on in Mexico. And by that time, by the next day, we had total genome sequences for 4 viruses from 3 patients from 2 states. And so that was the beginning of a very fast and furious 8 to 9 months since then. We also had put in place, thanks very much to my deputy, Dr. Dan Jernigan, a very robust diagnostic plan, and we were able to actually manufacture and distribute influenza diagnostic kits globally. And to date, we have distributed about 2,000 real-time PCR kits globally to about 153 countries, all 50 states, and DC and Puerto Rico. And as a result of this, we have received a lot of samples that are unsubtypable in the countries which have received these kits and are using them. Oftentimes, the unsubtypable samples are simply ones that are of poor quality, and we haven't really detected any novel influenza viruses, but we have seen in some cases information that indicates that there were dual infections, so we know that the possibility of reassortment is real. And then in addition, we distributed our regular WHO reference kits with antigens and antisera so that the labs out there can actually do HI identification of their isolates and do serologies. The development of a proficiency testing program is a work in progress, but we're hoping to work very, very closely with WHO to improve all aspects of proficiency testing and QA/QC in our WHO network labs. So as a result of being able to quickly deploy real-time PCR platforms and the real-time PCR kits to countries around the world, we have been able to get information and viruses even from many countries, particularly in EMRO and AFRO, from which we had not received influenza samples before. And just as an example, I'll list a few of the countries in Africa that have been sending us samples: Ethiopia, Nigeria, Kenya, Tanzania, Liberia, Uganda, Cote d'Ivoire, and others. And then we have countries in the Middle East that have been sending us samples, and many of our PAHO partners have also been sending us samples. And so we are developing a much more robust influenza surveillance network globally through our partnership with WHO. So what I'd like to do is just look at the timeline in a slightly different way, and I apologize if you can't see it very clearly, but here we have February 24th, the first case, actually single case, was detected in Mexico, and this was done retrospectively. So if we take this into account, and we know that we had this novel virus emerge during the time when Mexico was having its seasonal influenza outbreak, we can see that there was probably some time lost there simply because it's very confusing. It's like 1976 when we had the H1N1 swine influenza virus emerge during a time when we were having a lot of A Victoria virus circulate, H3N2 virus circulate. It was really very difficult to make sure that you weren't missing some of the H1N1 cases that might have been spreading from Fort Dix. And then I've talked about these particular timelines here, except that the first diagnostic kits were actually shipped to the state labs on April 29th. That's really in record time, and that's thanks to work with ATCC and especially with FDA for a very rapid turnaround of an EUA for use of this kit, of the swine influenza, H1N1 diagnostic kit. By May 3rd, the first diagnostic kits were shipped to the WHO network labs overseas, and on May 23rd, vaccine viruses were shipped to manufacturers. I hope I don't have to do any arm wrestling with Jerry over this timeline. We put this timeline together in conjunction with FDA. I'm not sure that all of the dates and all of the time intervals are exactly correct, but I just want to talk about something that I think is really, really important. And I think that what is really, really important is trying to move this point where the initial detection is actually closer to the event, closer to the emergence of the virus. And I don't think we can ever get the first case identified, but I think that we can certainly move this part of the timeline to the left. And I think if, for example, we could move it only 2 weeks to the left, which I think we could do today, simply because we have real-time PCR assays deployed in many, many more countries. And we could move this part of the graph where we're looking at vaccine availability, and I've used the U.S. as an example, but I think we could use many, many different country examples here for when vaccine was actually available. If we could move this part of the timeline to the left by 2 weeks, this part to the left 2 weeks, and gain a full month, just think of what it would have meant for our vaccination campaign in the U.S. We would have had vaccine available beginning September 5th instead of October 5th, ready to administer. And we would have been administering vaccine before the peak of illness. And I think that is a very important feature. I think if we look at events along this timeline, and I want to emphasize one particular area, and that is where FDA prepares the potency testing reagents, and where the vaccine manufacturers are actually able to make their clinical trial lots, judging how much vaccine is actually in each dose. I think that if we can make improvements in this particular area, and I know that China and some other countries actually did, but I think if we focus on this area as well, we can help move this, the availability of vaccines closer to when we really need them, and ideally prior to the uptick, the acceleration phase of the pandemic. There were some other issues that had to be dealt with during this H1N1 pandemic that I think are worth mentioning. Although we were able to ship to the vaccine manufacturers the vaccine candidate strains, some of those manufacturers were limited in their ability to develop the seed viruses due to the lack of facilities to grow the virus in large volume at the required biosafety level. So there were just a lot of different issues that really played into making this process a bit difficult. And in spite of that, I think we were very, very lucky, and I think we had many, many successes along the way. So the H1N1 response actually went, I would say, quite well. Some would say very well. We were better prepared than we had been in the past, and we were lucky. And I was thinking yesterday, reflecting on what the conversation yesterday would have been if we'd not had the preparedness exercises and investments due to the emergence of H5N1. It would have been a totally different conversation yesterday, and today probably as well. That we had diagnostic preparedness in the United States due to swine influenza infections in humans. We had the funding to produce and distribute the real-time PCR tests globally. The sharing of viruses and information accelerated vaccine development. It turned out that the H1N1 vaccine was quite immunogenic, and that the viruses remained homogeneous, so we didn't have to worry about multiple different vaccine viruses for different parts of the world and so on. The disease was relatively mild compared to Asian and Spanish flu pandemics, and vaccine adverse event surveillance has really not picked up signals. And so perhaps we can begin to lay to rest some of the GBS concerns. So instead of really talking about universal vaccines or movement toward universal vaccines, which is something else that's very near and dear to my heart, I just wanted to talk more about incremental improvements, and I've already mentioned them. But if we could really move the recognition of the outbreaks 2 weeks even to a month earlier, we would have had vaccine in a much more timely manner. And I think that we now have many more countries that can identify and subtype influenza viruses, and that are aware that unsubtypables raise the red flag for further investigation. And then if we have a focus on R&D that really tries to identify molecular determinants for high virus yield in eggs and cells, as long as we have to rely on the growth of the virus. This will really help us tremendously. And I know there's been some work done on this, but I think there needs to be a big push in this particular area, because as long as we're dependent on this technology, we need to improve virus yields. We have this problem not only for this pandemic virus, we're facing it for the H3N2 component for the southern hemisphere and perhaps for the northern hemisphere. And then we need to move vaccine production schedules to the left on the timeline, so that we can have vaccine available a bit earlier. And it could be in some pandemic situations you need it a lot earlier. So in addition, I think we need to focus on novel ways to quantitate antigens in older and new vaccines. So it doesn't matter whether we're talking about the egg-based inactivated vaccines or recombinant vaccines, we have to have ways to quantitate the amount of antigen in those vaccines. So just a couple of words about our avian influenza and pandemic influenza preparedness technical package, and this is something that we've been really working with WHO, DoD, and many other partners around the world to enhance international capacity for surveillance of influenza. And then we're trying, of course, to decrease the time to detect, report, and respond to cases. And I think one of the things that's been emphasized this morning in terms of regulatory preparedness, and certainly needs to be emphasized in terms of surveillance preparedness, is measurement and evaluation. So how do we really assess how much progress has been made, not for attribution, but more contribution in terms of can a country actually detect and respond to a new virus that spreads. We're talking about enhancing health systems preparedness, looking at the evidence base for vaccine introduction. I think a lot more will be said about that later. And of course, helping to implement vaccine policy where appropriate, all with the goal of decreasing the burden of influenza. Many of you in the room have probably seen this map, but others may not have. And because we had funding for H5N1 preparedness, we were actually able to put in place bilateral cooperative agreements with the countries that are shown in the colors there, in the green and blue colors. And so we've been working very closely with those countries and with the regional offices, WHO regional offices and headquarters to improve laboratory capacity. There's also a big effort to put in place research platforms and to use research platforms that are already in place, such as that in Bangladesh, to do specific studies, probe studies, and so on. And I think more will be said about that later on. We've also found that it's very important to have boots on the ground, and those countries where we have deployed CDC staff, and they're actually working hand in hand with ministries of health on the ground, we find we have the most progress. So I think I would disagree with David that it's too early to talk about lessons learned. But we certainly won't have learned all the lessons there are to learn at this point in time. So our preparedness and response for this pandemic would not have been possible without the funding and the resources and the efforts for H5N1 pandemic preparedness. The well-developed global networks, such as the Global Influenza Surveillance Network and the relationships and partnerships that were already developed, allowed for a more coordinated and measured global response. Rapid information and virus sharing was essential for this response, and we must preserve this rapid sharing so that we can develop better diagnostic tests and better vaccines. We need better surveillance for influenza in domesticated animals. This is essential for pandemic preparedness, and as difficult as it may be, we have to work with our agricultural partners and really emphasize the One Health concept. The H1N1 pandemic really reinforced our understanding of the two-way street involving exchange of influenza viruses between humans and domestic, sometimes companion animals. And as I said, there's much more to be done. We're hearing about H1N1 being transmitted from people to domesticated animals and many companion animals. And so we understand we may be creating additional reservoirs for influenza viruses. Having an influenza vaccine or having influenza vaccines 1 month earlier is likely to have made a big difference in disease reduction and vaccine acceptance, because if we look at the polls that are done on a regular basis at CDC, and we look at the vaccine-seeking behavior as reported by individuals in these surveys, we know that there was a much greater receptivity to receiving vaccine, and that people would be seeking vaccine more aggressively during the time that that H1N1 was circulating very, very actively. And as disease has waned, individuals are less interested in getting vaccine. So if we'd had vaccine earlier, there would have been a much better match between the desire to be vaccinated and the availability of vaccine. So for the future, we need to find funding for sustainable global flu surveillance. We need to continue surveillance and improving vaccine capacity building in all WHO regions, but as pointed out, especially in AFRO and EMRO. We need to be able to expand the real-time PCR assays for other respiratory pathogens. The next pandemic may be SARS or some other respiratory disease, not influenza. I already mentioned the next bullet point. And then I think also we need to have better signal-to-noise filters for respiratory diseases, because I think that we perhaps could have done a bit better in understanding that there was something important going on in Mexico. And then, as I mentioned, we need to streamline methods for measuring vaccine antigen content. There are many acknowledgments. It was really just a huge global effort, and we have learned our lessons from working with all of our partners. So just to remind you, we have a new player in the field, in the influenza field, the pandemic H1N1 virus, and it likes to reassort. So don't forget that this virus has that property, and that we also have H2, H5, H7, and H9 viruses circulating in domesticated animals. And the next pandemic may not wait another 40 years. Thank you very much.
Okay, thanks, Nancy, for that thought-provoking talk, particularly with respect to moving up the timeline. I think that's we'll get some comments and discussion on that. Our next speaker is Dr. Jerry Weir, who's Director of Viral Products at CBER. And he's, on short notice, graciously agreed to talk to us about what they do at FDA. Thanks, Jerry.
Thank you. As Nancy said, there's a lot of different areas of laboratory support that are critical for vaccine production. I was actually going to focus my short presentation on only one aspect, and that's specifically the development of potency reagents and that part of the process that's for vaccine development. So, this slide just shows the a list of the licensed influenza vaccines in the United States. And the point is that in the United States, just like the rest of the world, the vast majority of current influenza vaccines, whether seasonal or pandemic or pre-pandemic, are still inactivated split vaccines that are manufactured in embryonated eggs. We have one manufacturer of live attenuated influenza vaccines in the U.S., which is FluMist, manufactured by MedImmune. So, regarding potency of these vaccines, the potency of live attenuated vaccines is determined by essentially measuring the virus titer. It's actually, for the licensed vaccine in the U.S., this is called a focus forming assay, but it essentially measures how much live replicating virus is present. There is no standard that is involved in determining the potency for this because it's a single manufacturer and they have a validated assay. On the other hand, for all of the inactivated vaccines that are prepared by the different manufacturers, they all use an assay called the single radial immunodiffusion assay, or SRID. For this assay, this is dependent on the availability of standards to measure antigen content, which is the HA in the vaccines. The standards are specific for each vaccine strain, and the preparation and the calibration of those standards is a routine aspect of influenza vaccine manufacture and production. The current process of standards preparation and calibration takes weeks to months, and I actually don't have a timeline for you, so I hope you can remember the one that Nancy showed, and I think you saw some other ones this morning. It is a significant part of the process every year of making vaccines, though. But these standards for inactivated vaccines are necessary to ensure that multiple inactivated vaccines made from multiple manufacturers all produce the same clinical benefit. The next slide simply shows sort of a snapshot of what the SRID potency assay is. Okay, so this assay, it's an old assay, everyone knows that, but it's based on the diffusion of a virus antigen, which is either a detergent-disrupted virus or vaccine, through an agarose gel, which the agarose gel actually contains an HA-specific antibody. If you look on the bottom left of the slide, you actually see what one of these gels look like. This is a picture of the agarose gel, which is the antibody is embedded in that. The antigen is spotted on those little holes. It diffuses out. You get a ring where it precipitates, and the diameter of the precipitant ring is proportional to the antigen concentration. You use a standard curve to quantify the HA in the vaccine sample. On that particular example, you see antigen diluted down the gel plate, and you actually see one is an antigen standard, and then there's several vaccine standards next to it, and then the graph on the right shows that if you graph the log of the diameter versus the concentration, you get a linear relationship over at least some usable, workable range of antigen. So this is the SRID potency assay that's used by manufacturers. There's actually, in spite of the fact that it's an old assay, there's actually some strengths to this assay. One, it's actually not technically demanding, which is a good thing when it's being used multiple places in the world, multiple regulatory agencies. There's actually a fair amount of interlaboratory reproducibility. It does measure, and this is important, an antigenic form of HA, and the measured HA content does correlate with clinical benefit. All of these are strong reasons why this hasn't been replaced by a more modern assay. On the other hand, it has some serious weaknesses too. The reagent standardization is critical for having the assay to work, and it's time consuming. The assay is dependent, as I just said, on the strain-specific reagents. There is a limited sensitivity to the assay, in other words, the range at which the antigen can be measured. This has worked pretty well over the years, but as there's more and more of a push toward vaccines that may contain lower amounts of antigens, especially when complex with an adjuvant, this will be of serious limitation to the assay. And then last but not least, there are large quantities of reagents that are necessary for this assay needed to support worldwide vaccine manufacture. So I wanted to talk over the next couple of slides about the actual process of preparing and calibrating these potency reagents. Okay, as I said, a specific antigen standard and antibody standard is required for each vaccine strain, each strain that is in the vaccine. These are lyophilized and supplied together sort of as a kit, and there is a reference antigen which has an HA value assigned to it, and the antibody solution is a working dilution is provided for that, so that everyone that receives these has the value assigned to the HA antigen and a working dilution that they can actually use in the assay. The antigen standard is whole inactivated virus. There's actually two aspects of this. There's a primary standard that is prepared specifically for the calibration process, and then there's a secondary standard, which is the reference antigen. This is lyophilized, and this is what is supplied to manufacturers and other people around the world with a value assigned to that antigen. Okay, the antibody standard is typically hyperimmune sheep sera. Sheep simply because large animals get large volumes of antisera. And then the preparation of the antigen and the antibody standards proceeds in parallel. During this time frame that Nancy showed you a few minutes ago, all of this is going on at the same time. Antigens being prepared at the same time antibodies being prepared. And then these reagents are distributed by the WHO Essential Regulatory Laboratories, which includes CBRA at the FDA, but NIBSC in the UK, NIID in Japan, and TGA in Australia. Okay, so the calibration of the reagents, antigen reagents, is shown on this slide. The HA content of the primary standard that I mentioned in the previous slide, which is an inactivated virus and it's in a liquid form, is determined by physical-chemical means. What this means is that as each laboratory gets this primary standard, they determine the total protein. This is usually by nitrogen analysis or Lowry methods. And then they do calculate a percentage of that, of the percentage of hemagglutinin that is in that sample. This is done by SDS polyacrylamide gel electrophoresis and densitometry. There's actually a picture down on the bottom right of the slide that shows a typical gel. In other words, each laboratory runs this multiple times, scans it, takes an average, determines the percentage of HA in that sample. And then the HA value is assigned by using all of the WHO collaborating centers to come to a consensus on what the value is in that primary standard. The primary standard then, once that value is assigned, is used to determine a HA value in the large preparation that is the secondary antigen, which is the reference antigen. This is done using the SRID assay that I showed you a few slides ago. So the HA content of that standard is also assigned using through the collaborative network of the WHO ERLs. So they all work together for all of these antigens to assign the values before they are distributed. The reagents are actually distributed usually as soon as they are available, even before the values are assigned, such that they're available to use by the manufacturers as soon as the value has been determined by the WHO centers. The entire process, though, does take weeks. It's not a day-long thing. It takes several weeks to do this. As I said, at the bottom of this gel shows you a typical, just an SDS polyacrylamide gel of the type of procedure that is used for the densitometry. This slide shows or outlines what goes on to prepare the antibody standards, sort of a schematic at the bottom of it. But basically, the virus in question is digested with the enzyme bromelain to remove the HA, the hemagglutinin, from virus particles. The HA, which is now missing the transmembrane portion, is purified and then used to immunize animals. Animals are typically immunized multiple times in order to generate a hyperimmune sera. The working definition of when a sera is available and good for the SRID assay is a very empirical thing. It's basically animals are immunized, sera is taken periodically and used in the assay, and when it works, it's ready. These, as I said earlier, is distributed along with a reference antigen. The schematic at the bottom shows what I just said. The HA and the membranes cleaved by bromelain, purified, put into a sheet. So, we have a lot of current and future challenges to this process. First of all, we're all interested in improving the current process of reagent preparation and calibration. One of the things all of us would like to do is to expedite this whole process, shorten it from weeks and months down to days. This would be ideal. This actually could have a measurable effect on the timelines for vaccine production. In addition, it's very important that we all understand that backup procedures need to be developed to ensure reagent availability. I'll give you a specific example of that in a minute. But this is essential for emergence of a pandemic strain. You need to have procedures in place such that when something goes wrong or if something happens unexpected, you have a backup. There's also a serious need for developing new methods for potency determination. I mentioned earlier there is a need for assays that have greater sensitivity. These will become probably even more important in the future, and it would be ideal to have methods that are not always antibody dependent, because as I told you, the antibody process is always dependent on the amount of time it takes an animal to respond to make an antibody. And finally, last but not least, we need to develop new technologies to determine the potency of new generation vaccines. Cell culture vaccines are sort of the next step. These are already appearing. There's a question on the horizon right now of whether egg-based reagents that we have are suitable for cell-based reagents. This is something we're wrestling with right now. Adjuvanted vaccines, as I said, typically come with much lower antigen content, so we have to have assays that can measure this. On the other hand, there are other types of vaccines on the not too distant horizon that are fundamentally different than what we have now. DNA vaccines, but recombinant proteins, virus-like particles. Some of these vaccines may not even be just HA dependent, and so we'll need new techniques for developing. We need to develop new techniques so that we can assay the potency of these types of vaccines. Now, I'm going to give you a couple examples in the next few slides of ways in which, just examples of ways we're trying to approach some of these problems and challenges. This slide is titled Improving the Current Reagent Calibration Process. In the last year or two, there was a reported mass spec technique by some researchers at the Center for Disease Control, which uses an isotope dilution approach to quantify the HA in complex mixtures. We actually have now a multi-center collaboration, both with the CDC and with a couple of different centers at the FDA, not only CBER, but Center for Food Safety. And the purpose of this collaboration is to evaluate the use of this mass spec technique to determine the absolute quantity of HA in primary influenza standard. The little schematic the graph at the bottom just shows an example of a mass spec analysis of a complex mixture where you can identify the HA in the mixture and quantify it. And so what we're hoping is that if we can correlate these techniques, we could actually shorten the time necessary to determine the antigen content in that primary standard from weeks down to days. So this is an example of something we have underway. The second example I'll give you is developing alternatives to reagent production. As I mentioned, current techniques require the availability and the preparation of virus, specifically talking now about the preparation of the antibody standard. The HA, this requires that virus be available, that HA be purified before immunization to generate the strain-specific antibody. And the fact is, this is not always successful. We've had a couple of recent examples over the last few years. One, several years ago, on a particular strain of H5N1, it actually took several months for us to, not us specifically, but I'm talking about the global community, to figure out a way to purify the HA to put into sheep to generate an antibody. That was sort of a wake-up call that this could be a potential bottleneck. And the fact is, when the H1N1 emerged this past year, this became a serious problem. There was only one center in the entire world that was actually able to successfully generate enough of the HA by cleaving with bromelain to put into sheep to generate the antibody. Fortunately, they were successful and they supplied pretty much the world's needs. But it once again emphasized that this is a potential bottleneck and we have to have methods in place to address it should we have unforeseen problems in the future. And so what I've shown on the bottom of this slide is an example where we've used recombinant techniques to immunize animals and generate an antisera that would work in these SRID assays, showing essentially proof of concept, the feasibility that you can do it without even having the virus present. In other words, you can do it by recombinant means. And I actually have a couple of examples of where we generated antibody by traditional means and recombinant techniques, and then showed that they could assay the same vaccine samples successfully. And this last slide in this sort of group is titled New Methods for Potency Determination. As I said, this is something that we very much need, are new methods, and these are being explored by various centers and various manufacturers as alternatives to SRID potency assays. Some of the methods that are under development include ELISAs, but also mass spec, as I just mentioned, but also HPLC. And in fact, during this 2009 H1N1 pandemic emergence, we actually used HPLC techniques, several of the manufacturers, to formulate clinical trial material. This was, fortunately, a lot of these manufacturers had quite a bit of success and luck over the last few years of developing these techniques, and we were successful in using this for the initial formulation. I won't say that it's ready for prime time yet, but it was a good indication that there is some feasibility here, particularly in an emergency situation. The problem is that, as of yet, we've yet to show that any of these other techniques can actually measure an antigenic form of HA that can be correlated with clinical benefits. And it is also possible that standards may still be needed, although maybe not antibody for some of these techniques, which could, again, shorten some of the timelines. So in summary, influenza vaccines present unique considerations for product development, including the need to standardize vaccines from multiple manufacturers. Potency reagent development and calibration is time-consuming and is always a potential bottleneck in the production of inactivated influenza vaccines. Improvements are needed in the current process to ensure timely availability of sufficient supplies of vaccines, and the development of new techniques and methods for potency evaluation, as well as standards preparations, are needed for new generation vaccines, including some that are on the short-term horizon. Thank you, and I'll stop there.
Thanks, Jerry. Two excellent presentations. I have some questions, but I'll open it up to the audience first and see how that goes. Daniel.
Nancy, you talked a lot about some of the needs in surveillance. In thinking of the international perspective, where do you see some of the geographic gaps? In order to have a virus, you need to be able to detect unusual clinical events, obtain samples, test them close to the source, transport them to a more sophisticated laboratory to subtype them, determine whether it's a new subtype, isolate, etc. Where do you see globally in the GISN where some of the leading, the biggest gaps are, either in that chain or geographically?
I think it's on.
It's on. Okay. I was looking for the second and the third question, Daniel. Okay, so where are the gaps in that whole chain? I think for certain regions, and I hate to have us continually sort of single out Africa, but I think that's the region of the world where the least robust respiratory disease surveillance exists. And so there are challenges all the way along, from specimen collection, from identification of clusters of severe acute respiratory illness, because if we really want to, we might miss something, but certainly focusing on events where there are clusters of severe acute respiratory illness will help us greatly. we have to have the specimens collected and transported, perhaps not even tested near the bedside, but at least they have to be transported to a reference lab that's able to do real-time PCR and rule out or rule in certain pathogens. I think that we've made enormous strides, but in Africa, and you can see that as time, if you were watching the WHO website, you could see as time went by, we were really coloring in the continent of Africa. And there are also some Central Asian countries that still haven't reported detecting pandemic H1N1 virus. But if we, so there are some Central Asian countries, some countries in Central America where influenza surveillance and respiratory disease surveillance generally is improving very rapidly, but there's still gaps and needs there that we need to address. And then, of course, Africa. I think the Western Pacific Regional Office has really worked very, very hard on respiratory disease surveillance and has closed a lot of gaps. There's still some countries in that region that could use some additional help, some funds, focused funds, and so on. But the areas where we most need to concentrate are sort of Central Asia, some parts of Latin America, specifically Central America, Africa, and then a few countries in the Southeast Asia and Western Pacific regions. But if we look at where we were a number of years ago, we have made tremendous strides. One of the concerns that we do have is making surveillance sustainable. And as long as we can continue to provide the real-time PCR test kits, and we work with our partners to actually make sure that the PCR platforms are in place and that they can be used for multiple purposes, and that there are maintenance contracts for those machines and for the hoods and everything else that needs to be in place in a laboratory. I think it will be sustainable if we can demonstrate that and we can bring up a number of examples where by putting influenza capability in place, we were able to actually have everything that was needed for identification of an Ebola outbreak, Rift Valley fever outbreak, rapid response teams for those particular pathogens and so on. So again, the dual use sort of platform for surveillance and response, or multi-use platform for surveillance and response needs to be developed. And I think that influenza, because it's had so much attention and so much funding recently, can begin to provide that.
Rob? Yeah. Rob?
Rob Webster. I've been sitting quietly most of the meeting. Nancy and Jerry have identified, which in my opinion are the two major gaps in this area. The molecular biologists let us down most terribly in the preparation of high-growth reassortants. And we had to go back to Doris Bucher. Thank God for Doris Bucher, who used old-fashioned technology. Nancy is absolutely right. We have to understand high growth, and there is absolutely no excuse why we can't. We know that it's a polygenic property, and we have to characterize each of the subtypes, the requirements of the hemagglutinin, the neuraminidase, and the other genes. And my God, we can do it. We've got the technology. Let's put it into place. Let's BARDA fund that work, for God's sake, and get it done. Then turning to Jerry, the standardization of the vaccines, depending on one sheep in Britain, I believe it was. That's a catastrophe waiting to happen. We have got to move on from this old technology. Jerry has pointed the direction where we've got to go. We have to use that old technology, but to develop the new strategies of using the molecular techniques to produce the hemagglutinin, the end of the hemagglutinin, make the serum, show the antigenicity. And this is the other area that has absolutely got to be supported. This is the other big gap. And if we do both of those things, we can do what Nancy wants to do, reduce that timeline. So to me, those are the two big gaps that we've got to fill. They've both identified them. Let's get it done.
Yes, please.
Just a quick comment. First of all, in all fairness, Doris did a great job, though. And second of all, we usually have at least 2 sheep.
Well, I just would like to echo what Rob said. I mean, I think that we've learned a lot, and there's a lot of knowledge, but there's a lot of missing pieces, and we can do things better, but we need some concentrated R&D in these 2 particularly vulnerable areas. And so I think that these projects are very well worth funding. There are challenges, but I think we're at a point in time when we can make a lot of progress, and it is really our responsibility to do so.
So, yeah, and obviously BARDA is very interested in doing this, and we actually help fund some of the CDC studies on the mass spec approach, and we're very interested in the new approaches for getting vaccines early, quality vaccines early on. And the comments I had is that quality vaccines require quality virus samples from the minute it comes out of the nose of the person and goes into whatever you're going to grow it in. And so we have to deal with those regulatory challenges for making sure that you have quality virus samples. And one of the questions I have is, as far as I know, all of the manufacturers around the world of an H1N1 used the approved strains that came to them from WHO or the regulatory agencies that had been approved. And so one sort of question I have is there a time where we're talking about building vaccine capacity in developing countries where a strain could be generated in the country that would be approved for that vaccine manufacturer, which could conceivably save you a few days or a week, because you have to have the vaccine go through the regulatory, the virus seed go through the regulatory process, then go to the manufacturer. And I'm very happy with this paradigm. Or are we stuck with this paradigm where we're going to have this initial regulatory process and the process of getting the quality seed before we can start vaccine production?
Well, this is partly a WHO question. I mean, I think regulators as well as manufacturers around the world sort of rely on that WHO network to essentially provide them with a reassortment that has been undergone some oversight and from a center that has quite a lot of experience in making it.
Yeah, no, I tend to agree. I'm just throwing the question 10 years down the line, 15 years. Yeah. Is this something in terms of building capacity that we should look at, or just sort of assume that our vaccine production capacity begins with the receipt of an approved strain, I guess?
Well, I think it is clearly something that should be looked at, whether you could generate a high-growth reassortant anywhere at any time and use it, I don't know. I will say that a few years ago, I think pretty much the global community looked at the situation and said there were not enough centers actively trying to make high-growth reassortants, and I think there has been some improvement over the last couple of years. Our NIBSC colleagues got back into the business of producing reassortants. I think there's a little more capacity than there was even a couple of years ago.
Yeah.
But it's clearly an ongoing issue, though.
The other comment I had is that, you know, our vaccines are filled based on a 15 microgram. This time the decision was made that it would be filled based on a 15 microgram dose for all the manufacturers as the dose. And now we know that, you know, perhaps had we had the potency reagents early enough, we would have had a different potency of vaccine. Had we had the clinical trials early enough, we may have known that we could have used a 10 microgram dose, let's say. So we're stuck with this paradigm of the sort of the 15 microgram dose, where in essence we could have anywhere from 20 to 30% more vaccine if we had the better data early on to say how much vaccine would be filled do you think that's a possibility in terms of?
I think everything's possible. What you would not get around in that scenario is still the need for that clinical trial to determine that dose. And so I'm not sure if once you're committed to doing the clinical trial, like in this case,
what we had to do.
Yeah, how would you have shortened that much more? I mean, in a sense, in the U.S., we made the decision to go with a 15 almost before we had the final results from the clinical trial. I mean, remember everything was sort of going on in parallel. So I don't know. Would additional studies be helpful? Yes.
Well, yeah, and then the follow-on to that is, you know, off-label use of the filled vaccine. If the possibility of, if we really needed twice as much vaccine now as we have, you know, what is the possibilities, regulatorily speaking, of you know, off-label use of the vaccine?
Off-label use?
In other words, the physician decides he'll use half a dose in 2 people. Yeah.
Well, I don't think we ever end up recommending that.
You're right.
Other questions?
David's got a question.
Well, actually, just a comment on some of the discussion. I mean, in terms of the issues you're bringing up about the need to move forward in terms of standardization of some of the assays and methods for potency determination, I agree very much with Nancy and Jerry. I think we're now at a point where some of the new technologies are now sufficiently mature. I mean, Jerry says they're not ready for prime time yet, but I think we're actually close to that position. I think what we should be doing is a consolidated and coordinated review of those new technologies. I mean, China used HPLC to calibrate their clinical trial lots, and they gained a month, for example. So I think we are at a point where, you know, the science has advanced, where we can really try to push a proper evaluation, a full evaluation of those new technologies, and to see, you know, can they give us additional benefits over what we have already. So I'd very much support that. In terms of the capacity of laboratories to generate the vaccine seeds in the first place, I think, again, there has been some increase in that capacity, as you said, in the recent past. But I think it is something that is still, it's a vulnerability in the system. But we do need to be careful about, you know, just throwing it open to any and everybody to come up with a strain, because, you know, there are you know, it's a very complex process to develop a strain that is going to meet regulatory requirements, and so there are a limited number of laboratories that have that capacity. And while, yes, we can look to expand it on what we have, I don't think we should give the impression that we can just, you know, throw it open to anybody and everybody to come up with something, because I think we'll find ourselves in problems down the road with that.
Tony Colgate. I mean, the one hold-up with the H1N1 was waiting for the ferret testing to be done, because H1N1 caught us unawares. And with H5N1, the virus was going to be attenuated, and therefore there was a plan to ship the virus before the ferret testing had been done. With H1N1, we didn't really know whether the reassortment onto the PR8 backbone had attenuated it or not. So basically, there had to be a delay before the virus could be shipped to manufacturers to handle in facilities that were not BSL-3. So I think really the H1N1, again, we were all prepared for H5N1, and H1N1 came along, which kind of delayed us a little bit. Please, Nancy, go.
Yeah, I think the H1N1 did have some different features, but we did ship the H1N1 viruses to vaccine candidate viruses to manufacturers prior to the completion of the ferret studies. And I don't know if you remember the process, and I can't, recite the dates off the top of my head, but we were working with WHO very, very closely, having very, very regular teleconferences and working through the biosafety considerations and so on, and still getting the viruses out to the manufacturer so that those that had BSL-3 containment could begin to work on developing their own seed viruses. So I remember that very, very distinctly, and hope that we did the best to do as many things in parallel as possible, still recognizing that some vaccine manufacturers would be held back because they didn't have the level 3 biocontainment to do the work.
I mean, that's perfectly true. Most manufacturers didn't have BSL-3, and I think certainly we're trying to convince our senior management that we should have, and any help we can get on that would be useful. I mean, that is a lesson learned. I mean, it's not only the kind of the seed production, it is also the main egg production, which really can't be done in anything higher than BSL-2+, which is again what we were all set for, we all upgraded for H5N1, and then H1N1 came along and caught us unawares. Everything was done as quickly as possible, but I'm just saying there was a delay in us being able to start work on the seeds due to the ferret testing. Now, whether that's because we hadn't got our facilities ready or the virus wasn't shipped is another issue. It's just a kind of a general comment that that's something we need to address for the future and make sure that it doesn't hold us up next time.
So, well, Sid had a question, yeah.
I'm Sid from GPO Thailand. I would like to ask some more explanation from the floor. That are there any more standardization or the minimum requirement that CBER or US FDA have on the live attenuated vaccine? Since I think MedImmune also is one of the major produced vaccine that have been approved and used quite a lot in US. So how there's currently no standard of the live attenuated vaccine that I think US FDA should have some minimum criteria, or do you use WHO criteria for standardize this vaccine?
Okay, not positive I followed the question, but if we are you envisioning a scenario where we had multiple live attenuated vaccines, and whether we would need standards then for that? Is that the question?
Yeah.
Yeah.
Yeah, I think that's it.
Okay, so it's a little hard to speculate, but I'll take a stab at it. If, for example, we had a second live attenuated vaccine, and if it came from the Leningrad strain that is used in the rest of the world, my prediction would be that the vaccines would be so different that it would be hard to have a standard for those. But I'm just kind of talking off the cuff here. It's just not quite the same situation as the inactivated, where everyone does a version of, splitting the virus and wanting to standardize the amount of HA in it. I think if there were multiple live attenuated vaccines, we'd probably evaluate this on a case-by-case basis. If there was some standardization that one could use, it would obviously be a good thing, but it's just that I can envision that each attenuated vaccine would start with a completely different donor strain, and it might be very hard to have a standard for those. So I'm not sure I've answered the question, but...
Sorry, maybe not so clear, because I would like to ask that in case, even that there's no standard, do CBER or US FDA have any criteria that is the minimum that used for
okay. So yes, even without a standard, of course, for the licensed vaccine, the manufacturer has shown in clinical trials that a certain dose is effective against influenza, and once that's established in a clinical trial, then there are clearly specifications for how much virus the titer has to be in that vaccine. Yes, that's clearly established.
But FDA doesn't measure that, right?
It's not that clearly established. It's established for seasonal, and then
Right.
it is inferred that for pandemic you need the same dose, which we actually don't know.
That's true, though.
So, Joe, did you have I think you were...
Just a quick question. I'm still stuck on Joe Berzee from Flu Division. I'm still stuck on Daniel's question about what we need, and just maybe Nancy can amplify on her response, because I think Nancy's exactly right. There are clear geographic holes in our surveillance, and to say surveillance is fundamental in both pandemic preparedness and recognition of a pandemic, but also in making progress on the things that we've talked about here the last 2 days, and getting countries to actually use vaccine, I think seems trite at this point to say it's fundamental. I'm worried, though, that I'm equally worried that where the gaps are in Africa and Central Asia and some other parts where we clearly need more surveillance data, both to pick up a pandemic, to pick up the pandemic introduction to the country, or to make the case for seasonal flu need. There's an equal danger in letting slide the surveillance capacity that has been dramatically built up over the last couple of years because of the fears of H5N1. And Nancy, you have a longer term view of surveillance than I do, and I wonder if you would share that, because from my perspective is countries have engaged in surveillance for flu in increasingly so over the last 4 or 5 years because of the H5 pandemic and because of the specter of a pandemic, the H5 pandemic, the specter of a pandemic. We're passing through the pandemic now, or a pandemic right now. My concern is not only that we won't have surveillance in Africa that we need to build, but that the surveillance that's been established anew in many countries will be let go or will be decreased. And I think, getting back to Daniel's question about what we need to do, it may sound self-serving, I guess, but I think we need to support those countries that have nascent surveillance, that have juvenile surveillance systems just starting, to make sure they don't go away once the pandemic threat is also gone.
And Nancy, before you answer that, can I just add to Joe's question? I was going to ask a related question, but I can tag it to this. Is from CDC's perspective, are you, Abdullah Brooks again from ICDRB, sorry, are you seeing new partner countries getting the message about dual use or multiple use platforms and being committed to sustaining the newly established surveillance in those sites where they are? I mean, are you seeing real commitment to maintaining this beyond just the current pandemic in those countries that have started surveillance?
Yeah, those are very, very good questions, and I perhaps didn't express the fears that I have that are along those same lines, and being a realist and having worked in the area of flu for a very long time and seen interest ebb and flow, one of my concerns is that we will lose ground after the pandemic dies down and H1N1 becomes a routine pathogen that just circulates along with other seasonal influenza viruses. So I think that we had very short periods of time to speak today, but one of the things I wanted to emphasize more that I didn't really have time to do was this idea of partnerships, which I think have become ever more important, and the concept of platforms that are multi-use platforms, whether they're for surveillance or regulatory preparedness or whatever, become more and more important because we are in a world with an economic crisis. Certainly some parts more severely affected, but budgets are shrinking. And so we need to be very, very smart in how we partner, in how we use those platforms, and how we invest to make surveillance sustainable. And I think that what we've been trying to do at CDC is emphasize that we don't know what the next pandemic will be. It could be SARS. It could be something totally outside our area of our concept of understanding. Some new pathogen could emerge from some animal source, but if we are able to maintain these platforms, we will be able to detect those pathogens. Real-time PCR is a very versatile technique, and recognition of clusters of severe respiratory disease is really critical in all regions of the world, and we just need to make these systems sensitive and not lose ground. I think the global community can understand, based on this pandemic, just how quickly a virus can travel around the world. Any pathogen can travel around the world, and I'm sure we're not measuring the impact of H1N1 in sub-Saharan Africa at all. And I think that's, it's a pity we haven't been able to do it. Those teenage, very nascent surveillance systems hopefully will be grown up and through some very careful TLC, but it does take resources, and I think that's one of the things that all of us have to emphasize over and over again. There's some very focused projects that need resources, and we've identified some of those things today, but then there are a lot of these ongoing capacity building areas that need continued funding, and we just can't let them die.
Yeah, regarding this issue, I would like to share the Mexico experience to trying to maintain these new platforms that it has been very fast growing in the country. In Mexico, at the end of April, we have just one center, the National Center, the laboratory reference for episurveillance in Mexico, able to do real-time PCR. In the first month, we have 10 centers doing real-time PCR, and up to now, we have 35 centers doing real-time PCR. And we really need to maintain that. So one way we're doing is that dengue is a priority, for example, in Mexico. So what we've been doing is try to transfer the CDC Puerto Rico technology to do serotyping of dengue virus, virus surveillance of dengue with a different platform. We're standardizing this with this platform that we're using for influenza, and to have it for a different different network at the same time. So in this center, we can use it for another viral surveillance priority in Mexico, which is dengue. And this is very attractive to our authorities to give us more funding to maintain these platforms in the country. And we are going to try to do it for some other viral surveillance in the country to maintain them.
Thanks. Dr. Bloom? Yeah.
Just to pursue this discussion of what you define as a platform, there's a tremendous amount of foreign assistance in health for Africa for AIDS. Essentially 81% of U.S. ODA for health from the government goes to PEPFAR. And in a consequence of that, there are lots of support for laboratories in many countries, but particularly Africa, to do HIV. Very recently, they've been opened, amazingly, at this stage in the epidemic of tuberculosis, to be AIDS and TB programs, AIDS and TB clinical trials. So the question that I'm really asking is, do we need a laboratory platform for every single virus and every single infectious diseases? And since the platforms are both similar for many infectious diseases, either serological platforms or PCR platforms, and in my own view, much of PCR will be outdated in 2 to 3 years with non-thermal cyclers, cheaper things, and where everybody is moving is to point-of-care diagnostics, for which the Gates Foundation and others find at WHO are putting in a lot of effort. So the question I'm asking whether you can contemplate is, would it help the influenza effort to be part of a sustainable laboratory network for a variety of infectious diseases, or do you feel that that would compromise the expertise in any one? And if the former were the answer, then there's a start through AIDS and TB and malaria in Africa that one could build on.
Thanks very much for that comment. I perhaps didn't emphasize enough that we had really worked with the GAP labs, and so where it was possible in countries where the GAP labs were able to absorb some additional work, we were able to sort of just export primers, probes, the kit, and then people on the ground were able to actually do some of the testing. So it depends on workload, it depends on circumstances, it depends on country priorities and a whole variety of different things, but we did tap into that system very successfully, and I think that we will continue to do so where we can. We don't need, and I agree that we need to be thinking about next generation tests, point of care tests. You know, we didn't have enough time to really talk about all of the different diagnostic vulnerabilities that we have and where we might be going in the future today. But I agree, we need to be really thinking about next generation and how to find less expensive tests that can be used in the developing world.
Okay, I think we're out of time unless there are any burning issues. We'll break for coffee and be back in 30 minutes. Thank our speakers again. It's a great discussion. Thank you.